All right, so this lecture has to do with cells, their structure, function, size, types of cells. So by now in your educational career, you probably already know that there are two types of cells, the prokaryote and the eukaryote. Prokaryotic cells are basically defined by the fact that they really have no nucleus and they don't have any membrane-bound organelles. Eukaryotic cells are cells like what we have in our bodies, and they have their DNA, their genetic material, condensed in a nucleus, and they have several different types of membrane-bound organelles. An important term for cells is apoptosis. Apoptosis is the cell death. Sometimes apoptosis occurs because of the fact that cells are not healthy and they need to be destroyed. Perhaps the DNA, when it was copied, it wasn't copied correctly. But it's actually very important for a lot of other different reasons too. The tissue between your fingers and toes while you're developing as an embryo, that tissue needs to be basically destroyed so that you have your fingers and toes and they don't have that webbed tissue between them. So it requires apoptosis or cell death of those cells. The reabsorption of a tadpole tail, that's another example of apoptosis that's actually necessary and healthy for an individual. Cancer cells we're going to learn later on, these are cells that divide uncontrollably, and one of the reasons that they're able to do that is they override that apoptosis programmed cell death, and so they're able to keep dividing and keep surviving. Apoptosis often occurs when digestive enzymes from other cell organelles like the lysosome, those things are released into the cell, and those digestive enzymes, once they're released out of the lysosome into the cell, it's just going to basically break down those cell components until the cell itself dies. Endosymbiosis. This is an important term and an important theory, and it describes how eukaryotic cells came to be as they are with our membrane-bound organelles. It's believed that at some time there was an ancestral cell that basically took in another, perhaps a bacteria, some other cell that was aerobic. So that means it's using oxygen in order to create its energy. And so that bacteria or that other cell was taken in and it survived. It wasn't digested as food, it wasn't broken down, it wasn't destroyed. That cell was taken in and it survived. And so now this cell right here is using oxygen to make its energy, makes more energy than what it needs, and so those extras are able to be used by the ancestral cell. So it's a benefit for both of those cells. That eukaryotic cell keeps that mitochondrion or keeps that cell, and then as it grows and divides and reproduces, then that cell stays in there and stays part of it. Chloroplasts are thought to also have been brought into eukaryotic cells the same way. So an ancestral eukaryotic cell pulls in some other maybe bacterial cell that's using light energy to create its food and create its energy, and so it pulls that in as well. That survives in here, and then now we have a eukaryotic cell that has mitochondria and chloroplast. Of course, that would probably be a plant cell or a bacterial cell, whereas an animal cell would have the mitochondria only. So within cells, there are lots of different types of organelles. Scientists have learned about organelles through cell fractionation. Cell fractionation is a process that they can study that cell structure where they break up the cells and they place them in a centrifuge. As they spin the centrifuge, the heavier stuff is going to go to the bottom, and then they can separate off whatever portion is at the top. Then whatever was at the top, they run it through the centrifuge again, spin that, take the heavy stuff off the bottom, take the liquid portion off the top, spin it again, and they continue to separate the components of the cells that way. So organelles that have internal membranes That's a benefit for them because it gives them more surface area. So instead of just being surface area right here, now this cell has all of this surface area around where all of these membranes are at. So because it has more surface area, that means it has more places for reaction to take place because there's increased locations for those reactions to take place. So kind of think of it Thanksgiving. You need to make Thanksgiving dinner. You have a very small kitchen with a very small counter. Well, if you don't have a whole lot of counter space, then it's going to be very hard to make a whole lot of dishes for Thanksgiving. You might be able to make a couple of dishes and that's about it because that's all the space that you have. But if you have a kitchen and the kitchen itself is still small, but you have counters that go all the way around the walls, now you've got a whole lot more space and you can make more different dishes for your Thanksgiving dinner. What kind of organelles that cells have lead to a specialization of that cell. So for example, since mitochondria, the powerhouse of the cell, since mitochondria are making the ATP, if there are cells that have a large number of mitochondria, they're going to have a lot of energy reserves. So this could be something like a muscle cell. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, because these have to do with packaging and shipping and making proteins and moving proteins around in the cell. If a cell has a large amount of these types of organelles, then they're going to be responsible for making and shipping a lot of products within that within the organism. Absence of a nucleus. Well, if a cell doesn't need a nucleus, it's not going to go through the energy to create one. Erythrocytes, red blood cells, they don't have a nucleus. And that is a cell that's specialized to carry oxygen. And remember, its surface area and its shape is the kind of donut shape that makes it flexible. And so it is more important for a red blood cell to carry oxygen than to carry genetic information. Presence of a chloroplast, you've got a plant cell that can be autotrophic. A large flattened cell, this is going to have lots of surface area, and so that's going to be really good for digestion or absorption. A large number of ribosomes. Ribosomes make proteins, so those cells are going to be responsible for making important proteins for the organism. There are definitely limits to cell size. Cells, we know, are naturally small. You got to use a microscope to see most cells. The upper limit of eukaryotic cells are 10 to 100 microns. Technically, an egg, like a chicken egg or an ostrich egg, those are the largest eukaryotic cells that we have. And of course, we can see those without a microscope, but you know, they're a very specialized type of cell. There are also some algae that are eukaryotic cells that are pretty large as well. You can hold them in your hand and you can see them without a microscope. But generally speaking, cells are going to be microscopic. There are limits to cell size. Surface area to volume ratio is a very important limit to cell size. The surface area to volume ratio depicts the fact that cells have to be a smaller size. So a ratio, remember, has to do where you're comparing two amounts, and that ratio shows the comparison of one amount to another, one quality to another one. So for example here, if I've got 20 students in the class, the ratio of boys to girls is 5 to 15, 5 boys to 15 girls, but you can also simplify that. So 5 divided by 5 gives me 1, 5 divided by 15 gives me 3. So 5 to 15, this is the same ratio. So in this class of 20, I have 1 boy for every 3 girls. As the cells get bigger, their volume, the internal volume, increases faster than the surface area. But all of that volume within that cell, it still needs to have all of the nutrients from the outside, the surrounding environment. Because of the fact that smaller objects have a greater surface area to volume ratio, it's much easier for them to get the nutrients that they need into the entirety of the volume of their cell. So for example, I have 1, 2, 3. I have 3 cells here. Notice that each one of these cells have the exact same volume. So the volume of the cube, the volume of this rectangular object, the volume of this rectangular object are all 64. However, when I look at surface area, the surface area of this cube, length times width times 6, is 96. The surface area of this cube, so I've got length times width, and I've got 2 like this. Length times width, I've got 2 like that. Length times width, I've got 2 like that. So once I find my surface area and add it up, I get a surface area of 112. So the volume is the same between this cell and that cell, but this cell is more likely to survive than that one because it's got more surface area. There are more places on the surface of this cell for nutrients to be able to diffuse into the internal part of the cell. For this cell right here, the volume is 64, just like the rest of them, but my surface area, I've got length times width, I've got 2 of those. Length times width, I've got 2 of those. Length times width, I've got 2 of those. When I find my surface area and add it up, the surface area for this one is 160. So there is a lot more surface area on this cell. So of the 3, this one's actually more likely to survive than those others because of the fact that its surface area to volume ratio is greater than any of the others, because the surface area is much larger than the ratio of any of the others. And I don't think I put that on here, but the surface area to volume ratio of this one right here, this would be 1 to 5 to 1. So 1.5 to 1. The surface area to volume ratio of this one would be about 1.75 to 1. So 1 3/4 to 1. The surface area to volume ratio of this one would be 2.5 to 1, 2.5 to 1. So this one, 2.5 to 1 compared to the original 1.5 to 1, it's a much larger surface area to volume ratio. So that limit is because of the fact that of those metabolic requirements, because of the fact that you need to get every nutrient into every part of this internal part of the cell. So if it's larger, then it's harder to get everything into each internal part of the cell. So what do we need to do in order to solve this problem? Make it multicellular. So the organism itself can still get bigger, but the cells can stay a smaller size that's more effective to getting their nutrients. Of course, you're going to have other challenges if it's multicellular, but this helps solve that surface area to volume ratio. Example of that, so these are the same 2 cubes. So this cube right here, it's got a surface area of 4 by 4 times 6, because there's 6 sides, 96. The volume of this cube is length times width times height, so it's 64. Here, instead of being a solid cube, I have 8 cells. 1, 2, 3, 4, 5, 6, 7, 8. I've got 8 individual cells. So the surface area is each cube's surface area, 2 by 2, and it has 6 sides. Then I have 8 cells, so my total surface area is 192. The volume is 2 by 2 by 2, and I've got 8 cells, so 2 by 2 by 2 times 8 gives me 64. So once again, it's the same volume, but a much greater surface area when I have individual cells rather than just one big cell. Surface area to volume ratio for my solid cube, 96 to 64, which is about 1.5 or 1.5 to 1. Surface area to volume ratio for my multicellular organism, 192 to 64, which is 3 to 1. I just doubled the surface area to volume ratio simply by having multiple cells, and it ends up being the exact same size as my original cube. So having multicellular organisms is a benefit because I can keep my cells small so that the surface area to volume ratio is reasonable enough to help the cell survive, but then I can keep adding more and 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 more little cells so the organism itself can continue to grow bigger. Surface area to volume ratio is a very important factor in survival.